Hello there, EP. We are going to finish talking about evolution by finishing up with the history of life on Earth. So, the Earth is very old. It's about 4.6 billion years old, but for the first billion years, there was no life on Earth. And so it was really just this place of intense volcanic activity. There were a lot of this different mixture of gases in the atmosphere, but not any that we'd be used to now. So there were different things like water vapor and nitrates and sulfates and carbon dioxide, but not the oxygen and the nitrogen that we'd expect now. So it is theorized that life arose because those gases, or from those gases, small molecules were able to be synthesized. And so these would be things like amino acids and nitrogenous bases. And so with those being out there, they would start to kind of come together, forming larger things like proteins and nucleic acids. And this resulted in the formation of the first protocell, which is basically a membrane-like barrier with stuff inside. And then as soon as the first self-replicating molecule formed, and it's thought that this is RNA, and that got packaged inside one of those protocells, that was kind of like the first cell. And so that is how they thought life arose. And so the reason it's thought that it was RNA is because RNA is so versatile compared to DNA. It can be an enzyme, referred to as a ribozyme. It has all these different functions, but it also can be replicated similar to DNA. And so this theory was created by Operin and Haldane, and they had this idea that, all the starting material for making amino acids and proteins, nitrogenous bases and nucleic acids were present in Earth's early atmosphere. And in addition to that, there was a lot of energy that was found in the atmosphere. So lightning, UV radiation. And so the energy was being used, the light energy from lightning, energy from UV radiation was being used to create these very simple organic molecules, leading to the creation of the first cell. And so they referred to that, the mixture, I guess, of all these gases as this primitive soup. And here's an example of that first protocell and some of that RNA. And then finally, Miller and Urey came through and they actually tested Operin and Haldane's hypothesis. And so they basically put all this mixture of gases that were thought to be in the Earth's early atmosphere into this apparatus, and they used electricity as the energy source. And what they found is that they were able to synthesize amino acids. And so it's likely that this is how those first simple molecules are synthesized, and it led to the development of larger things, those macromolecules like proteins and lipids, nucleic acids. All right, so we've talked about fossils just a little bit, but there's just a few things to add. I think you all know what fossils are. And so looking at these, the fossil record kind of dates back to when, well, not exactly when life was first arose, but we have those simple prokaryotes in the fossil record that kind of come first, followed by the very simple eukaryotes, those unicellular eukaryotes, about 2 billion years after that. And then over here we have much more complex eukaryotes, including this first kind of fish-like eukaryote. And then finally, here's a very famous fossil. It's called Tiktaalik, and it is basically thought to be the fish that first colonized land, so most likely leading to the land-dwelling vertebrates, or the evolution of the land-dwelling vertebrates. And then finally, some more complex, this is thought to be, even though it looks kind of dinosaur-like, it's thought to be mammal-like fossil, and then a reptile-like fossil. And so fossils, again, can be used to as a piece of evidence for evolution, but the other thing that they can be used for is figuring out kind of the relative age of something. And so there's two ways to use fossils to figure out age. The first one I will show you a picture of in just a minute is referred to as relative dating. And it's basically using like the order that the fossils were laid down as a way of gauging kind of relative to other fossils how old something is. Because the ones that are found more towards the bottom should have been laid down first, and then other newer ones kind of came on top of them. This second one is radiometric dating, and this is basically using radioactive isotopes to figure out the relative age of something. And so what we can use here is half-life. And so a half-life is the number of years it takes for 50% of a sample to decay. And so radioactive isotopes normally kind of decay at this constant rate, and we can figure out that rate. And so what we can do is we can use those known half-lives to compare the amount of, substance A to what it turned into substance B, or what it decayed into substance B, and figure out how old these different samples are. So I will go through a few sample problems with you. At any time, feel free to pause, try to work out that problem, and then I will help you out with the answer. All right, so here is that relative dating. So you would expect something more towards the bottom to be much older than something more towards the top, because obviously it was laid down way before the stuff at the top. And so here's that other example. Again, this is the older stuff followed by the much younger things. All right, and then here's that half-life. And so I guess now, you could say, for any of us, 100% of the carbon in us should be, or the radioactive carbon, I guess, should be in the form of carbon. And then basically what this is saying is that it takes a certain amount of time, which is referred to as one half-life, for half of that radioactive carbon to convert into radioactive nitrogen. Might be the other way. And then it takes another half-life for half of this half to convert. So about a quarter of that original would be left. And then another half-life for half of that half, or half of the quarter, an eighth to convert. And so it's basically like taking a piece of pie and then cutting it in half. So the first person gets half, and then I only have half left. So the next person that comes along gets a half of that half, so a quarter of the pie. Now I only have a quarter left. The third person that comes along, they get half of the quarter that I have left, which is an eighth, and so on and so forth. And so it's basically like each time half of the pie is being converted, or half of this sample is being converted into a different form. So I have a few practice problems for you. So the half-life of carbon-14 is 5,730 years. And so it's saying if the amount of carbon-14 is 1/8 of the ratio of present day, how old is this sample? And so the first thing I always do when I figure these out is how many half-lives have passed. So after the first half-life, there'd be half the carbon left. After the second, there'd be a quarter of the carbon left. And after the third, there'd be an eighth. And so what this information is telling me right here is that there has been 3 half-lives. So then what I take is I take those 3 half-lives, I multiply it by this 5,730, and my answer is 17,190 years. All right, so here is a second problem for you all. And so in this problem we have potassium-40. It has a half-life of 1.3 billion years. And it's saying when comparing this, we have 1/4 the current ratio. So again, figure out how many half-lives. Take those number of half-lives and multiply it by the amount of time of one half-life. And so you should have gotten 2.6 billion years. All right, this one has quite a few questions, but anyways, it's saying we have 160 grams of something, and we have 15 years is the time of one half-life. So after 15 years, you'd expect there to have been one half-life, and so half of that sample should have converted. And so there should be 80 grams of the sample left. After 30 years, that is now 2 half-lives, and so half, or a quarter of the sample should be left. So we should have 40 grams. And then after 60 years, that's 4 half-lives. So half of a half of a half of a half, which I believe is a 16th. And so we should have 10 grams of that sample left. And then finally, this last one, it says the half-life of an isotope is 2 years. How long would it take for 4 milligrams to decay into 0.5 milligrams? So again, in this case, figure out how many half-lives have passed. And so for me, I thought about the fact, well, after 1 half-life, there should be 2 milligrams. After the second, there should be 1 milligram. And then after the third, there should be 0.5 milligrams. So there's been 3 half-lives that have passed to decay the 4 into the 0.5. Each half-life is 2 years, so 3 times 2 gives me 6 years total. All right, so just one last time, here's that picture of the half-life. So again, think of a pie. So half of that pie, after the first half-life, half of the pie has been converted into something else. After the second half-life, now half of that half that I've left, so about a quarter of the pie is left. Now I only have a quarter left, and so half of this is converted, so about an eighth of that pie is left. And then finally, don't forget I have just an eighth left, so half of that is converted, and I just now have a teeny tiny sliver, a 16th of that pie. All right, so the last thing I am going to mention, and I think it's just a slide or two more, is this geologic time scale. So when looking back through time, they can't just count in years. There's just been way too many years. And so they kind of divide it up into these different groupings. And so this largest grouping is an eon. And so you'll notice we are currently in this eon here, followed by an era. So we're currently in this Cenozoic era. Period, we're in the Quaternary period, and then an epoch, and we're in the Holocene. So the Holocene is kind of current time. But we can look back and look at, the Jurassic time, which I'm sure you all know of, and things like that. So that Jurassic period is when there were a lot of dinosaurs. And then finally, putting this into a clock analogy, looking at those huge kind of developments over time. We have the origin of Earth, where we first have those prokaryotes appearing, followed shortly after that by abundance of oxygen, free oxygen in the atmosphere. And so that's part of the reason why prokaryotes don't or tend not to use oxygen. It just wasn't present when they were evolving. Here we have the eukaryotes evolving, multicellular eukaryotes. You'll notice animals, followed shortly after by that Tiktaalik and the colonization of land. And then way here at the very, very end, we have humans. So compared to the history of Earth, we've only been here for a very, very short time. And then one more way of looking at this, again, Earth way back here, prokaryotes came first, followed by the simple eukaryotes, the much more complex eukaryotes. Somewhere in here, I have it somewhere, was that oxygen. And then diversity of life increased, and we'll talk about that tomorrow or next time I see you. Colonization on land, and then again, we would be way down here at the very end. All right, so a few reminders. Do not forget to turn in your Following the Global Gene through time on Google Classroom. And make sure you are finishing up your Unit 1 review in that review packet. And then finally, I will be available for questions if you have any today from 9:30 to 11:30 and 1:30 to 2:30. I will send out a link later. See you all soon.